The fundamental unit of life. Do you know what is the fundamental unit of life? It is cell. Cell is called as the fundamental unit of life. Do you know who discovered the cell? It's Robert Hooke. In 1665, Robert Hooke observed a thin slice of cork under his own microscope. He found small box-like structures. They were like small rooms of a honeycomb. Robert Hooke called these boxes as cells. The meaning of the word cell is little room. So cells are the building blocks of all living things. That means all living things are made up of cells. Certain organisms are made up of only one cell. Means the whole body of the organism is only one cell. Such organisms are called single-celled or unicellular organisms. Uni means single. Examples of unicellular organisms are bacteria, amoeba, paramecium, and chlamydomonas. Some organisms are made up of many cells. Such organisms are called multicellular organisms. Multi means many. Examples of multicellular organisms are plants, trees, animals, and human beings. All cells of a multicellular organism develops from a single cell. Cells produce new cells of their own kind by cell division. All cells come from the pre-existing cells. So here we learnt three important points about this cell. These three important statements are collectively called as cell theory. Let us see what are those. 1. All living organisms are composed of cells. This means that whether an organism is a single-celled bacterium or a complex multicellular organism like a human, all life is fundamentally made up of cells. 2. The cell is the basic structural and functional unit of life. That means cells are the smallest unit of life that can carry out all the processes necessary for an organism to live. Cells are the building blocks of life and perform various functions depending on their special job. 3. Cells arise from pre-existing cells. This statement is given by Rudolf Virchow. It tells that the new cells are formed from pre-existing cells through the process of cell division. These three principles are collectively called as the cell theory. We learned that multicellular organisms are made up of many cells. Is the whole body of a multicellular organism is made up of similar kind of cells? No. Multicellular organisms are made up of different types of cells. Now, let us see the different types of cells of our body. Muscle cells, blood cells, nerve cells, bone cells, ovum, sperm cells, fat cells, etc. Why these cells are in different shape and in different size? The shape and size of these cells depends on the function they perform. You know that in our body we have different systems to perform different tasks. For example, heart pumps blood, liver secretes digestive juices, kidneys filter the blood, and brain controls the body. This means that in our body, different jobs are done by different parts, just like how different people in a factory does different jobs. We call this as division of labor. We will observe this division of labor inside a cell also. A cell is made up of different parts and each part performs a specific function. Now let us see the different parts of a cell. Generally, a cell has three main parts. They are cell membrane, nucleus, and cytoplasm. Cell membrane or plasma membrane. Cell membrane is also called as plasma membrane. Plasma membrane protects the inner components of these cells, just like how our skin protects our inner parts. Plasma membrane controls the entry of materials into the cell and also controls the exit of materials from the cell to the outside environment. It works like a checkpost between two different states. You know that at the checkpost, checking of vehicles and verification of licenses is done. In the same way, cell membrane verifies the materials and decides whether to allow them inside or not. It permits only the selected materials. For this reason, cell membrane is called selectively permeable membrane. Now, let us see how the exchange of materials takes place between the cell and its surroundings. The exchange of gases takes place between the cell and its surroundings by a process called as diffusion. Do you know what is diffusion? The movement of molecules from a higher concentration area to low concentration area is called diffusion. Here we can see the concentration of oxygen outside the cell is high compared to the concentration of oxygen inside the cell. So the oxygen molecules, they move from higher concentration to lower concentration. That means these oxygen molecules, they enter the cell through the plasma membrane. This process is called diffusion. There is one more phenomenon through which the transfer of water molecules takes place between the cell and its surroundings. It is osmosis. The movement of water molecules from a place of lower solute concentration to a place of higher solute concentration through a semi-permeable membrane is called osmosis. Let us understand osmosis with a simple activity. Take some raisins. Raisin is covered by a thin semi-permeable membrane and it has a thick sugary pulp inside. That means the raisins have high concentration of sugars inside. Now take a beaker with some water. Now drop the raisins in this water. The concentration of the sugar in the water is less compared to the sugar inside the raisin. So the water in the beaker is called a hypotonic solution. Now osmosis takes place and the water molecules move into the raisin through its semi-permeable covering and the raisins swell up. This is called as endoosmosis. Now take another beaker and prepare a thick sugar hypotonic solution. The concentration of the sugars should be higher than the concentration of sugars in the swollen raisin. Now place the swollen raisins in this beaker. The water present in the raisins comes out into the hypotonic solution and the raisins become shrunk. This is called exoosmosis. So this activity shows us that in osmosis, water molecules move through a semi-permeable membrane from a hypotonic solution, that is lower solute concentration, to hypotonic solution, that is higher solute concentration. Then how do the transfer of other materials takes place into the cell? For the entry of some materials into the cell, energy is required. This kind of transport is called active transport. Now let us see the composition of the plasma membrane. Can we see the structure of plasma membrane through a compound microscope? No. The structure of plasma membrane can be seen only through an electron microscope. Plasma membrane is made up of carbon compounds like lipids and proteins. Plasma membrane performs a special function in amoeba. Organisms like amoeba engulfs the food materials through their cell membrane. This process is called endocytosis. Cell wall. Now let us see the structure and function of cell wall. Both plants and animal cells have cell membrane, but plant cells have one more covering over the cell membrane called as cell wall. Cell wall lies outside the plasma membrane. Do you know with which material the cell wall is made up of? The plant cell wall is made up of cellulose. Cellulose is a type of complex carbohydrate. It gives structural strength to the plants. Sometimes plant cells undergo a condition called as plasmolysis. Do you know what is plasmolysis? If the water present in a living plant cell is lost through osmosis, then the contents of the cell shrink away from the cell wall. This phenomenon is called plasmolysis. Importance of cell wall in plants. Cell walls in plants, fungi and bacteria help them handle watery environments without bursting. Plant cells absorb water when placed in diluted surroundings, causing the cells to swell. As the cell swells, it presses against the cell wall, creating pressure. The cell wall pushes back with equal force, balancing the pressure of the swollen cell. Unlike animal cells, plant cells can tolerate big changes in their surroundings due to the presence of cell wall. Nucleus. Now let us look at the structure of nucleus. The nucleus is covered by a nuclear membrane. This nuclear membrane has two layers. The nuclear membrane also has pores. These pores are called nuclear pores. Materials enter and exit the nucleus through these pores. The nucleus has chromatin inside. Do you know how the chromatin looks like? It just looks like a tangled thread. But it changes into rod-like structures called chromosomes during cell division. Chromosomes contain DNA and protein. Let us look at an example to understand the functioning of chromosomes. Your phone has software to control its functioning. In the same way, every cell has DNA to control its functioning. Our phones have different apps to execute different tasks. In the same way, DNA has different genes to execute different tasks. The functional segments of DNA are called genes. That means DNA is made up of genes. Genes are the building blocks of DNA. Do all cells have a distinct, that means clearly defined nucleus? No. In certain single-celled organisms like bacteria, the nuclear membrane is absent. The nuclear region is undefined and contains only nucleic acids. Such type of nucleus is called nucleoid. The organisms whose cells lack a nuclear membrane are called prokaryotes. Pro means primitive or primary. Karyote or karyon means organism. Then what are the cells with a clear nucleus called as? Eukaryotic cells. Are there any other differences between prokaryotic and eukaryotic organisms? In prokaryotes, besides the absence of nuclear membrane, the membrane-bound cell organelles are also absent. On the other hand, the eukaryotic cells have nuclear membrane as well as membrane-enclosed organelles. So by this, we understand that prokaryotic organisms have poorly organized cell organelles compared to eukaryotic organisms. Cytoplasm. We have learned that cell is covered by cell membrane. Do you know what is inside the cell? The space inside the plasma membrane of a cell is filled by a semi-transparent fluid. This semi-transparent jelly-like material is called cytoplasm. What does this cytoplasm consists of? Cytoplasm consists of water, minerals and enzymes. It is the place where many biochemical reactions takes place. There are so many organelles floating in the cytoplasm. These organelles are called cell organelles. These cell organelles keep the cell alive by performing different tasks. Each kind of cell organelle performs a specific function, such as making new materials in the cell, clearing up the waste material from the cell, and so on. Let us look at structure and functions of cell organelles. This is smooth endoplasmic reticulum, rough endoplasmic reticulum, ribosomes, Golgi apparatus, lysosomes, mitochondria, plastids, and vacuoles. Each of these organelles performs a specific function. Let's see some short details of each of these organelles. Rough endoplasmic reticulum. This is called as protein factory because it makes new protein molecules. Smooth endoplasmic reticulum. This is called fat factory because it makes new fat molecules. Golgi bodies. They are called dispatch centers because they handle the packaging and distribution of materials that are prepared in the endoplasmic reticulum. Mitochondria. It's called powerhouse of the cell because it produces the energy from the glucose. Vacuoles. They are called storage bags. They store different materials. Lysosomes. They are called suicide bags because they contain the digestive enzymes. These digestive enzymes digest the foreign materials like bacteria and damaged cells. Plastids. They are called pigment stores because plastids contain different pigments. These are the special cell organelles that are found only in plant cells. Plastids also help in photosynthesis. Now, let us see why do cells have different cell organelles. Let's find the answer to this question with the help of an example. We cannot play football, cricket, basketball, and tennis all together in the same court. Different courts are required to play different games. Similarly, in a cell, a lot of chemical activities takes place. Different chemical activities require different conditions and machinery to take place. All reactions cannot take place in the cytoplasm, so they need separate organelles. To keep these chemical activities separate from each other, cells use membrane-bound organelles. These organelles are called cell organelles. Can we see these cell organelles under a compound microscope? No, we cannot see the cell organelles under a compound microscope. But cell organelles can be seen with the help of an electron microscope. Let us study the structure and functioning of cell organelles in detail. Endoplasmic reticulum. Now let us learn about the structure and functions of the endoplasmic reticulum. First, let us locate the endoplasmic reticulum inside the cell. Yes, this is endoplasmic reticulum. We can see that it is located around the nucleus. This looks like a network of tubes and sheets. It is made up of some special membrane. This membrane of endoplasmic reticulum is similar in structure with the plasma membrane. The endoplasmic reticulum is of two types. This is rough endoplasmic reticulum and this is smooth endoplasmic reticulum. The rough endoplasmic reticulum is like a network of sheets folded around. There are so many dot-like structures present in the rough endoplasmic reticulum called ribosomes. Due to these ribosomes, it has a rough appearance. So that's why endoplasmic reticulum with ribosomes is called rough endoplasmic reticulum. Now, let's see the functions of rough endoplasmic reticulum. Rough endoplasmic reticulum makes new protein molecules with the help of ribosomes. That's why rough endoplasmic reticulum is also called as protein factory. Functions of smooth endoplasmic reticulum. Smooth endoplasmic reticulum appears as plain sheets and tubes. Smooth endoplasmic reticulum synthesizes or makes new fat molecules. What happens to the new protein and fat molecules made by the endoplasmic reticulum? These proteins and fats are used by the cells to make new cell membrane. We know that cell membrane is made up of lipids and proteins. So, for the formation of new cell membrane during cell division. These two materials are required. The process of formation of new cell membrane is called membrane biogenesis. Some proteins and fats are converted to enzymes and hormones and are secreted out of the cell to execute various functions. Apart from protein and lipid manufacture, what are the other functions of endoplasmic reticulum? Endoplasmic reticulum helps in the transport of materials between various regions of cytoplasm. It also allows the transport of materials between cytoplasm and nucleus. 2. It provides surface for many important biochemical reactions to take place. 3. In vertebrates, smooth endoplasmic reticulum plays an important role in detoxifying the toxins and drugs. So, these are the structural features and functions of endoplasmic reticulum. Golgi apparatus. Now, let us see the structure and functioning of Golgi apparatus. This is Golgi apparatus. Let us observe its structure. It looks like flattened bags or vesicles. These vesicles are arranged parallel to each other and form stacks called as cisterns. Now, let us see the functions of Golgi apparatus. In a factory, the machines that manufacture the item and the machines that pack them are different. Once items are made, then they are sent for packing and dispatch. In the same way, in our cells, protein molecules are manufactured in the endoplasmic reticulum and then they are sent to the Golgi apparatus for modification, packing and dispatch. The proteins that are manufactured by the endoplasmic reticulum are modified and stored in the Golgi apparatus. When needed, they are packed and dispatched in the form of vesicles. Golgi apparatus performs three important functions. 1. Storage of materials. 2. Modification of materials. 3. Packaging and dispatch of materials. The Golgi apparatus gathers simple molecules and combines them into more complex molecules. Then it packs these big molecules in vesicles. Golgi apparatus may store these vesicles for later use or dispatch them out. These vesicles are delivered to different cell organelles inside the cell. Sometimes these vesicles are delivered outside the cell to the target location. Since these cell organelles were discovered by Camillo Golgi, they are called as Golgi apparatus. Lysosomes. First, let us locate lysosomes inside the cell. These are lysosomes. Let us see the structure and functions of lysosomes. Lysosomes are sac-like organelles. The lysosomes are formed from the Golgi apparatus. Lysosomes are covered by a membrane outside and filled by a fluid inside. This fluid contains digestive enzymes. Now, let us see the functions of lysosomes. Their main function is to clean the waste and unwanted materials inside the cell. They work as waste disposal system of the cells. What kind of waste materials are found inside the cell? Sometimes a bacteria, virus, or some undigested food particle that enters the cell. These things are considered as a waste and they are to be removed. Sometimes the parts of the cell are damaged or worn out. These parts are also considered as waste or unwanted materials. These kind of materials are cleared by lysosomes. Sometimes if a cell is damaged, the cellular components try to repair it using materials like proteins and fats that they get from endoplasmic reticulum. But if the damage is beyond the repair, lysosomes help the cell to self-destruct. Lysosomes burst and the digestive enzymes digest the components of the cell. That is why lysosomes are called as suicidal bags of these cells. Mitochondria. These are mitochondria. Let us see the structure and functions of the mitochondria. Mitochondria are double membrane organelles. Each mitochondria has two membranes, one outer membrane and two inner membrane. The outer membrane is porous. That means it has pores. The inner membrane is deeply folded. Do you know why the inner membrane of mitochondria is deeply folded? Mitochondria need more surface area to carry some special reactions in which a special energy-rich molecule called ATP is released. So, to provide more surface area, the inner membrane is deeply folded. Now, let us see what are ATP. ATP means adenosine triphosphate. These are energy-rich molecules. In our cells, many reactions takes place and all these reactions need energy. And in our cell, there are so many new materials are produced. So, for the production of these new materials, ATP are required. That's why ATP are called as cell currency or energy currency of cell. Generally, in a cell, nucleus has DNA and it passes the information to the cell organelles like endoplasmic reticulum to make proteins necessary for cellular activities. That means all the cell organelles follow the instructions of the nucleus. But here, in case of mitochondria, it is a bit different. Mitochondria, they have their own DNA and ribosomes, so they can manufacture some of their own proteins. That's why mitochondria are sometimes called as strange organelles. This is all about mitochondria. Plastids. Now let us see the structure and functions of the plastids. These are the cell organelles that are found only in plant cells. Plastids are of two types. 1. Chromoplasts. 2. Leucoplasts. First we see about chromoplasts. Chroma means colored. The plastids that have color pigments in their stroma look colored and are called as chromoplasts. The plastids that contain chlorophyll and carry out photosynthesis are called as chloroplasts. So chloroplasts are green in color due to the presence of chlorophyll. Some chloroplasts contain yellow or orange colored pigments along with the green colored pigment chlorophyll. Now we will see the second type of plastids, leucoplasts. Leuco means white. So leucoplasts are colorless, that means white plastids. Their main function is storage of materials. Do you know what are the different materials that are stored by the leucoplasts? Starch, oils, and protein granules are stored by the leucoplasts. Externally, that means from the outside look, plastids look similar to mitochondria. If we observe the internal organization of a chloroplast, the innermost membrane is folded and arranged in stacks called as grana. The space between the grana is filled by stroma. Just like mitochondria, plastids also have their own DNA and ribosomes and can make their own proteins. Now, let us see about vacuoles. Vacuoles are these sacs for storage of materials. Materials in the form of solids and liquids are stored in them. In animals, the vacuoles are in small size, whereas in plant cells, the vacuoles are in very big size and occupy 50 to 90% of the cell volume. How do bigger vacuoles help the plant cell? The vacuoles in the plant cell are filled with cell sap. They help the plant cell in two ways. 1. Provides turgidity and rigidity to the cell. 2. They store many important materials like sugars, amino acids, and various organic acids and some proteins. In unicellular organisms, vacuoles perform some important functions. For example, in organisms like amoeba, the food enters the food vacuoles, where it gets digested and absorbed. In the same way, contractile vacuoles are formed to collect and excrete the excess water and waste materials. This is all about vacuoles, cell division. Now we will see the process of cell division. Cell division is the process by which new cells are formed from pre-existing cells. Organisms need new cells for growth, repair of their damaged tissues, and for reproduction. Growth means adding new cells to the body. So, in growing organism, continuous cell divisions produce new cells. Sometimes our cells may get damaged. To replace the damaged cells, new cells are required, and these cells are formed by cell division. For the process of reproduction, organisms need to produce new reproductive cells or sex cells. These cells are also produced by cell division. Now, let us see the different types of cell division. What are the two different types of cell division? There are two different types of cell division, that is 1. Mitosis, 2. Meiosis. First, let us see mitosis. Most of these cells are divided by this mitosis. This type of cell division helps in the growth and repair of body. For example, a boy grows into a man. For this, his bones and muscles are to be grown. Growth in the bones and muscles means formation of new bone and muscle cells. These new cells are formed by the mitosis. In the same way, for repair and maintenance of our body, new cells are required. For example, if someone falls and gets their hand or leg skin bruised, in a few days, we see new skin is formed. This is because of mitotic divisions of the skin cells. Do you know our skin loses 30,000 to 40,000 cells a day? All these cells are to be replaced with new cells. So, these new skin cells are formed by mitosis. In mitosis, one parent cell divides into two daughter cells. These two daughter cells are identical and have same number of chromosomes as mother cell. For example, the number of chromosomes in a mother cell are 46. The daughter cells also will have 46 chromosomes each. But for the process of reproduction, some special cells are required. These special cells should have half number of chromosomes of the parent cell. So, to achieve this, reproductive cells are produced by a special type of cell division called as meiosis. The main purpose of meiotic divisions is to produce reproductive cells or gametes. Now, let us see how this is different from mitosis. In meiotic division, the parent cell divides into two daughter cells that has the same number of chromosomes as their parent. That means if the parent has 46 chromosomes, then the daughter cells also have 46 chromosomes. But this is not end of meiotic division. These two daughter cells divide further and produces 4 daughter cells. These daughter cells will have half the number of chromosomes as compared to the mother cell. That means if the mother cell has 46 chromosomes, finally the daughter cells have 23 chromosomes each. So, in meiotic division, the number of chromosomes reduces to half. The sperm cells formed in the testes of males and egg cells formed in the ovary of females are formed by meiotic division. So, the gametes or sex cells have half the number of chromosomes compared to the other cells of our body. Why do sex cells have half the number of chromosomes? This is because during fertilization, the male gamete with 23 chromosomes fuses with female gamete with 23 chromosomes and forms a zygote with 23 pairs of chromosomes. That means 46 chromosomes. This zygote further divides by and develops into a baby.